Hello, my name is Bertrand. This is my biology teachback for the Princeton review, where I'll be discussing the nervous system. The nervous system consists of billions of neurons, which are nerve cells, and these neurons carry electrical signals known as impulses all throughout the body. As you can see, I have drawn a rough sketch of a neuron. Now, like most cells, neurons have a centralized nucleus and a cell body, and the cell body, is referred to as a soma. You may have also noticed these projections along the cell body. These are referred to as dendrites. This much longer projection going along this way is known as the axon, which then ends at an axon terminal. Now, at the start, I had mentioned that these neurons carry impulses, which are electrical signals. So how are these impulses carried? Well, these projections, the dendrites, receive an impulse from a neighboring neuron, and then they transmit that impulse to the cell body and through down the axon, where the impulse then ends at an axon terminal. So what is this impulse? Well, a neuron that isn't experiencing any impulse is referred to as polarized, which means this neuron is at rest and has its resting membrane potential. It is termed this way because there is an electrochemical gradient. This means that it is more positive outside of the membrane than it is on the inside of the membrane. Now, this electrochemical gradient is established by two main proteins. Now, the first protein is referred to as a sodium-potassium ATPase. Now, this protein is a channel that uses ATP as an energy source to pump out 3 sodium ions to pump in 2 potassium ions. Now, the other pump is referred to as a potassium leak channel. Without these ions cannot cross through the membrane. They need a channel to help do this. Now, because there are so many sodium ions being pumped out and so many potassium ions being leaked out, and both of these ions are positively charged, it leaves the inside of the neuron very negatively charged. So this is its resting membrane potential, which is typically minus 70 millivolts. Now, there's what are referred to as voltage-gated channels in the membranes of neurons. Now, these voltage-gated channels open when the cell membrane reaches a particular voltage. In this case, it's when it reaches negative 50 millivolts. Now, when this occurs, there's an influx of sodium and an efflux of potassium. This, as you can imagine, changes the electrical potential of the neuron from its resting membrane potential to something else, known as the threshold potential. Now, following this threshold potential, these voltage-gated channels begin to open, causing an influx of sodium and an efflux of potassium from the axon. Now, this causes an action potential. Graphically, as we can see, the very first sign at number 1 is the sodium channels are open, which means that sodium begins to rush into the cell. Next, at number 2, the potassium channels open, meaning that the potassium begins to leave the cell. This part of the graph is referred to as depolarization. Now, when an action potential is being depolarized, it goes from minus 70 up to positive 40. Next, in the repolarization phase, the first step to happen at number 3 is that the sodium channels become refractory, which means there's going to be no more sodium entering the cell. So, as we continue down to number 4, potassium continues to leave the cell, which causes the membrane potential to essentially restart, so to repolarize. since it was polarized when it was at rest, it is going back to the polarized state. Next, at number 5, the potassium channels are now closed. Also, at this point, the sodium channels are becoming reset. Now, when repolarization occurs, it always overshoots. It always goes past the resting membrane potential of -70. This is referred to as hyperpolarization. When this occurs, the extra potassium outside diffuses very quickly, bringing it back to this resting state, as we saw here. So because of this, this is what occurs in the axon. During an action potential, the electrochemical gradient changes, becoming more positive on the inside than on the outside, because it goes from -70 millivolts to +40 millivolts. Now, one of these action potentials happens in roughly 2 milliseconds, and they are all along the same speed. So you might be wondering, what happens to an impulse when it reaches the end of an axon, to the axon terminal? Well, when it reaches the axon terminal, the impulse is transferred to another dendrite of another neuron or to an organ. The point at which they meet is referred to as a synapse. So as you can see here, this will be our first neuron, our initial neuron that we were discussing here. This is the axon terminal of our first neuron. It is neighboring the dendrite of neuron number 2. So as you can see here, what's happening is the impulse, the electrical impulse, is turned into a chemical impulse. Now these chemicals are known as neurotransmitters. The most common neurotransmitter is acetylcholine, so I will use acetylcholine for this example. The neurotransmitter acetylcholine is carried within these little bubbles known as vesicles. Now, these vesicles, they fuse with the edge of the membrane of the axon terminal and release their contents in a process known as exocytosis. Now, once the contents are released, they enter, which is known as the synaptic cleft. The synaptic cleft is the area in between neuron 1 and neuron 2. So as the acetylcholine, or whichever neurotransmitter we have, is saturated and floating around this area, it is attracted to receptors, acetylcholine receptors, on the second neuron, the dendrite of the second neuron. So all these dendrites here have receptors that would accept the neurotransmitter from their neighboring neurons. So when this occurs, the signal has been passed from an electrical signal to a chemical signal back to an electrical symbol. Once the neurotransmitter binds the receptors, sodium channels open, and that influences the sodium to rush in, causing an influx of a positive charge within. This then causes, like before, the neuron to depolarize, which is enough to transmit the action potential down the second neuron. And the whole cycle begins. So quickly, just to recap, here we have a neuron. The neuron has an electrochemical gradient. When this electrochemical gradient is disrupted by an influx of sodium and an efflux of potassium, then it goes from its resting membrane potential to its threshold potential. This then influences the ion-gated channels to open. The first one to open is sodium, causing a depolarization effect. Halfway through the depolarization, the potassium channels open. This then brings us up to a positive 40 megavolts. When this occurs, the sodium channels close, repolarization occurs, and then the potassium channels close. There's a little extra potassium left over, giving us this hyperpolarization, but they quickly diffuse away and we are back at our resting membrane potential of minus 70. As this occurs, the action potential travels along the axon down to the axon terminals. Now at this point, the impulse is still an electrical signal. However, it is transmitted into a chemical signal when vesicles containing neurotransmitters such as acetylcholine are released at the axon terminal into the synaptic cleft through a process known as exocytosis. The released neurotransmitter and acetylcholine is then attracted to receptors on the dendrite of a neighboring neuron. When this occurs, the chemical message is then again transferred into an electrical message where these gated ions open, causing another influx of sodium, and the action potential can progress. Thank you so much for listening. I really hope you had a chance to learn something, and I'll see you again. Bye!